This is lecture 3 on general senses, olfaction, and taste or gustation for Biology 178. This is also a lovely picture of some delicious curry from Taste of the Himalayas. There are locations in Carlsbad and San Diego. It's a Tibetan style curry, so it's like a mix between Thai and Indian. Mwah! Enjoy. So when discussing sensory receptor morphology, sensory receptors are always located at the peripheral end of a sensory neuron. They're going to be exposed to the outside environment. They come in three types of morphologies: a simple receptor, a complex receptor, and then a special sense receptor. Simple receptors are exactly like they sound like. They are free nerve endings. Now, they may be myelinated or unmyelinated. They're exposed to the outside environment and respond with general touch. An example would actually be a nociceptor or a pain receptor, as these free nerve endings, once stimulated, will activate the entire receptor going towards the spinal cord. Complex receptors are sensory receptors that are enclosed in a specialized connective tissue. An example here would be like a Pacinian corpuscle in the skin that senses touch. It has specialized connective tissue to kind of filter information that goes through it. Finally, there's special sense receptors. Now, these are specialized nerve cells or receptors that release neurotransmitters onto a sensory neuron at a synapse. So there's different examples of this. One of the big ones is going to be a hair cell in the ear. This is a specialized mechanoreceptor that intakes the information and sends that information to the actual neuron to be transmitted. The same thing could be true of a photoreceptor in the eye that's going to break down and interpret photons before transmitting that onto a sensory neuron. Now, the area monitored by a sensory receptor is called the receptive field. The size and density of the receptive fields dictate how sensitive you are to a stimulus in that region of the body. For example, your fingertips are going to have a very high density of sensory fibers. This means that they're going to be more sensitive to things. However, if we have the receptive field on, say, your abdomen, the skin of the abdomen, it's going to be less sensitive. Same thing as your tongue would also be very sensitive. Now remember that the larger the receptive field, the poorer the ability to localize a stimulus. So you're not going to be able to detect the 2 points of discrimination between them as well. Now, before we get too far into the special senses, we're going to talk about general sensory receptors and their different classifications. We have nociceptors, thermoreceptors, chemoreceptors, and several types of mechanoreceptors. We're going to be going through each one. Now, photoreceptors are a type of general sensory receptor, but it's always considered special sensory, and we're going to talk about that more later. So, our first few types. Thermoreceptors. These are going to be temperature receptors. We have free nerve endings of these that are located in the dermis and skeletal muscles. We also have free nerve endings in the liver and the hypothalamus as well. These are going to detect variations in temperature, both high and low, and help the central nervous system to make coordinating responses. Chemoreceptors respond to water and lipid-soluble substances that are dissolved in body fluids. We have a number of receptors that are going to be either in the brain, detecting the different compositions of cerebral spinal fluid, going to be in the hypothalamus, reaching into the blood vessels to detect the differences in gas concentrations in the blood. And we also have different receptors in, say, the kidneys, or that detect different interstitial fluid to detect the different salinity of that fluid and determine whether or not we actually need to absorb more water or absorb more salts. So chemoreceptors are going to detect different chemical compounds in fluid. Mechanoreceptors respond to distortion of their cell membranes by opening or closing membrane channels. So they're going to respond to different stretching, compression, or twisting, and they're going to contain gated ion channels that will respond and open or close in response to this distortion. Now we have three main types. The first type is a proprioceptor. Proprioceptors monitor the positions of muscle and joints. The muscle spindle that we talked about previously is a fantastic example of a proprioceptor. They are the most structurally and functionally complex of general sensory receptors. If you really want to understand how a proprioceptor works, let's go through a little exercise right now. Close your eyes and put your right hand above your head. Open your eyes and now look up. Your right hand is above your head. How are you able to do that? It's because you could actually feel the position, the muscles, and the joint structures of everything within your right arm to your right hand and how it got above your head. You could compare where that hand was in relation to the rest of your body. That feeling of where your hand and where your arm are in space is based upon the information that is obtained from proprioceptors. Baroreceptors are another type of mechanoreceptor, and baro means pressure. These detect pressure changes in the walls of blood vessels and portions of the digestive, respiratory, and urinary tracts. One of the major baroreceptors that we have is actually found in the carotid artery. It detects the changes in blood pressure throughout your cardiovascular system, and it allows you to make different changes like enlarging the blood vessels, vasodilation, or shrinking the blood vessels down, vasoconstriction, in order to help maintain a homeostatic blood pressure throughout the system. Tactile receptors are the final type of mechanoreceptor. They provide sensations of touch, pressure, and vibration, and they can be extremely sensitive, either detecting fine touch and fine pressure, or very crude, just detecting overall pressure on the system. You have many tactile receptors in the dermis of your integument, as well as in the tissues of your tongue. The hair cells in your ear also are tactile receptors and mechanoreceptors that detect sound and in the form of vibration. So they're going to deal with vibration. Another type of receptor class that we have are photoreceptors. We'll briefly go over this now and then talk about it in a later lecture. But photoreceptors are sensitive to visible light, specifically the light particles called photons. In some animals, it's actually these photoreceptors are actually sensitive to infrared or ultraviolet light, so they can see things in an entirely different spectrum than we can. Now, they are always considered a special sense receptor, and in humans, photoreceptors are limited to rods and cone cells. Rods would detect different light intensities, so see everything on a grayscale, whereas cones are going to detect color. And these, of course, are going to be in the retina of the eye. Nociceptors are another type of general sensory receptor class. They're going to be simple receptors, and these are receptors that respond to the damaging stimuli that are interpreted by the brain as pain. So they are pain receptors. They're a very unusual class of receptor, as different types respond to different modes of damaging stimuli. So there's different temperature, mechanical, or chemical within the nociceptor class. And some are only stimulated when actual tissue is damaged. This is called a sleeping nociceptor. So nociceptors come in multiple types, and that's an important key aspect to understand. Now, there are two broad types based upon speed and different classifications under these, but we're only going to talk about the two major types. The first type is a type A fiber. These are going to be faster and myelinated. And that's because they carry sensations of fast pain. Think of an injection from a needle or a deep cut, and it is highly localized, so you feel it where it happened, so that way you can identify it. A great example I like to give of this is actually a paper cut, where it gives you an immediate, sharp, painful response to become aware of this area. Type C fibers are going to be the opposite of type A. They are slower and unmyelinated. They carry sensations of general burning or hard to localize pain. So it's going to be in that general area. Oh, it feels like it's kind of over here. And that's going to help you just kind of be aware of the pain in that region, but you can't really localize it as well. Another way to classify receptors is based upon their response. Receptors can either be tonic or phasic. Tonic means that they are always active. They either increase or decrease action potentials as the stimuli change. So if we increase the stimulus, this increases the frequency, and if we decrease the stimulus, this decreases the frequency. Phasic are going to normally be inactive. They become active only once the different conditions that are present are strong enough to elicit an action potential here. Phasic responses usually show adaptation. Once the stimulus reaches a steady state, they turn off. This allows the body to ignore information that has been evaluated and not found to be a threat to homeostasis. A great example of this is when you initially put on a t-shirt, is you're going to put on a shirt, you feel that the shirt is on you, and then after a while, you don't even notice that the shirt is on you anymore. We're now going to begin going through the different special senses of the body, including smell, taste, hearing, equilibrium, and vision. The first that we'll talk about is smell, otherwise known as olfaction. Olfaction uses the olfactory organs, which are paired structures that provide this sensation. They are located in the nasal cavity on either side of the nasal septum. The inferior surface of the cribriform plate, the superior portion of the perpendicular plate, and the superior nasal conchae of the ethmoid bone all help to house the olfactory bulbs that are going to deal with this sensation. Here we can actually see the olfactory bulbs as they extend through from the brain. This, of course, is going to be cranial nerve 1. And we have the olfactory receptor cells that are going to extend through the cribriform plate of the ethmoid bone and into the superior nasal conchae, where they are covered by an epithelial layer. Now, your olfactory organs are composed of 2 layers. First is the olfactory epithelium. This contains olfactory receptor cells, which are modified neurons. Each one of these cells is going to form a knob that projects past the epithelial surface and into the surrounding mucus. There are different supporting cells that are going to help to facilitate the receptor cells, the action potential through them. And then there are going to be basal cells. These are stem cells, and they help to constantly produce new receptor cells. Now, this is one of the few examples of neuronal replacement in the entire body. And that's because that's what allows us to smell something, to get away from it, to refresh our palate, as it were, and then smell it again. We need to essentially exchange those cells out over time as they become used. The next layer is the lamina propria. Remember that a lamina propria is going to be a layer of kind of connective tissue, like a thick layer of glue. Here it consists of areolar tissue, different blood vessels, and nerves, and also will house the olfactory glands, things like Bowman's glands. The secretions absorb water and form a thick pigmented mucus in this region that will extend out and around the epithelium. So if we take a look at the upper right corner there, this is what we're going to be looking at, is that olfactory epithelium. And now we're going to zoom in on that portion. Here we can actually see the difference between the olfactory epithelium and the lamina propria. The olfactory epithelium will, of course, house the supporting cells, and then the major olfactory receptor cells that will have dendrites that extend into the actual mucus. From there, we have different stem cells, or the basal cells, that will divide out from there, and that would connect and fuse directly to the lamina propria, which affixes it to the actual bone above. You can see the olfactory gland that would be inside the lamina propria that would release mucus to go out and around from there. The thing that we smell is called odorants. Odorants are dissolved chemicals that stimulate the olfactory neurons. They interact with membrane receptors called odorant binding proteins on the membrane surface. Generally, these are small organic molecules. The strongest smells are typically associated with molecules of either high water or high lipid solubility. As few as 4 odorant molecules can activate a receptor cell. So smell by itself is a very sensitive sense. We can detect things of a very low frequency. I'm now going to go through the process of olfaction and the actual physiological steps that allow you to smell. Understand that when you initially smell, you inhale something through your nose, you're actually going to have that air circulated around and it becomes more turbulent. This more turbulent air is going to warm it and kind of stir it up, like how you see people stir around a chemical or a glass of wine before they smell it. That's going to kind of mix up the odorant molecules. Once they're mixed up and in our nasal conchae, they will go all the way up into the actual olfactory epithelial mucus. Once there, they will stimulate and bind to a receptor protein in the actual dendritic surface. From here, that will activate a G protein. That G protein is going to allow adenosine triphosphate to convert to cyclic AMP. This is done via the actions of the enzyme adenylate cyclase, which was triggered by the active G protein. Now, understand that cyclic AMP is effectively a communicator. All it is letting the cell know that something needs to change. In this case, CAMP, cyclic AMP, is going to bind to a sodium channel that is closed. By binding to this channel, it will thereby open it and allowing sodium ions to go into the cell. Remember that allowing sodium ions to rush into the cell will cause it to become more positive, thus depolarizing. If sufficient depolarizing occurs, if enough of these sodium channels are open, then we actually generate a full action potential, and that will activate the cranial nerve, thus allowing us to interpret smell. We will now move on to gustation, which is the sense of taste. This provides information about foods and liquids that we eat and drink, and it is interpreted by gustatory receptors, otherwise known as taste receptors. The most important ones are going to be distributed over the superior surface of the tongue. They are also found in adjacent portions of the pharynx and larynx, so the back of the throat and the top of the windpipe, and epiglottis. The number of taste receptors that we actually have decreases with age. However, we can maintain quite a bit of taste receptors if we continue to use them. This is where you have different chefs or professional tasters that continually work and practice the skill of tasting. Gustation itself is heavily dependent upon a structure called the lingual papillae. Papillae, remember, means nipple-shaped mound. Now, the lingual papillae are epithelial projections on the superior surface of the tongue. These are what people most commonly associate with being taste buds, but they're actually not. These are going to be the protrusions on your tongue that would house the taste buds or gustatory receptors inside of them. There are 4 types of lingual papillae. 3 of them actually contain taste buds, whereas 1 of them is simply structural in nature. The first type is velate papillae. These are going to be very large and contain the most amount of taste buds that we have. They have about 100 taste buds per papillae, and they form a V near the posterior margin of the tongue. The next type is folate papillae. These are found on the lateral margins of the posterior region of the tongue, and they also contain taste buds. Fungiform papillae have a mushroom shape, hence their name being based off of fungus or mushroom. Each papillae contains about 5 taste buds, and they are found on the anterior 2/3 of the superior surface of the tongue. The final type of papillae actually does not contain any taste buds. This is the filiform papillae. Filum means thread. These are going to provide friction and help move objects around in the mouth. They are found on the anterior 2/3 of the superior surface of the tongue. Here we can see examples of each one of the type of papillae that we talked about. Starting with the velate papillae, you'll notice how large they are. They house a number of taste buds in there. We have the folate papillae, much smaller, going to house taste buds. The fungiform, notice its mushroom-like shape with a few taste buds protruding. And the filiform papillae has thread-like projections, almost sharp in nature. And that's not going to have any taste buds, but it does have neural receptors in it to allow it to move correctly. Now this is going to be an electron microscope look at these colorized images. Here we can see the fungiform papillae. We can see filiform all around it that kind of gives our tongue that hair-like appearance and allows us to grip things. We can see an example of a velate papillae towards the back as well, with a nice thick overall papillae structure on top, openings around the side that allow it to have chemicals dissolve and activate the taste buds deeper beneath. Now, remember I said the filiform papillae are going to be very thread-like and sharp. This is because they act as a sense of Velcro. Now, this is a cat tongue, where we have the filiform papillae of the cat tongue. If you've ever been licked by a cat, it feels like Velcro is licking you. And that's because the cat tongue is covered in more filiform papillae than almost any animal out there. It's a lot of feline species that have this. This is why they can use their tongues to actually clean their own bodies, because they have sharp little threads all across their tongue that allow them to pull things off of their body. Now we're going to talk about taste buds, which are going to be housed in the velate, fungiform, and folate papillae. Each adult has approximately 5,000 taste buds, and they're composed of gustatory cells, basal cells, and then sensory receptors that are attached. Gustatory cell is a taste receptor cell. These extend slender microvilli or taste hairs into a surrounding fluid through a taste pore, which is simply a narrow opening. Typically, these cells survive about 10 days before being replaced. You have about 40 to 100 receptor cells per taste bud. Now, the basal cells are going to be very similar to what we talked about in olfactory senses, and that's that they are stem cells that divide to produce more gustatory cells, so they are continually replaced. In the upper right corner of this picture, we can see a wonderful diagram of a velate papillae taste bud. So here we can see the taste pores, which are going to be openings on the side that has microvilli extending out. What these will do is actually try to grab the different sorts of chemical compounds that secrete down into the tongue or into these kind of taste pores. From there, it will interpret that information via the, as you can see, reddish pink gustatory cells in this diagram. That will then transmit the information via the axons of the sensory neuron all the way back down into the different nerves of the central nervous system. We can see different diagrams of taste buds and how they would actually transmit this information into the central nervous system as well. We have 4 primary sensations that these taste buds will actually detect: sweet, salty, sour, and bitter. The taste buds throughout the tongue actually provide all 4 sensations because there's no difference in taste bud structure that detects these. They all can detect all of them. However, there is a sensitivity to these sensations across the long axis of the tongue for many individuals. Past the 4 tastes of bitter, sour, salty, and sweet that we already discussed, we have 2 other sensations. The first is a pleasant, savory taste called umami, which is a Japanese reference to savory. Examples of this would be beef broth, a French dip sandwich, or Parmesan cheese, truffles, mushrooms that have been sauteed down a little bit. So that very strong kind of meaty taste. These receptors are present in the velate papillae. It's a more unique taste that seems to combine a lot of essences, but is kind of strong on its own. The other type of taste are water receptors. This is going to more be about the desire for thirst, of quenching that thirst and intaking water. These are typically concentrated in the pharynx, and the information from the water receptors are sent to the hypothalamus. And this is going to affect the water balance and blood volume regulation, and also prevent the over-ingestion of water. While all taste buds have the ability to taste sweet, salty, sour, and bitter, your taste buds are 100,000 times more sensitive to bitter and 1,000 times more sensitive to sour compared to sweet and salty. This may have a huge survival component. Toxic compounds are often bitter, whereas acids are typically going to be sour and can create chemical burns in the body. Overall, our sensitivity with these declines with age. This is why when you're younger, you might hate the taste of coffee, but as you get older, you tend to be able to tolerate it more. This is also why maybe some of those sour candies that you thought were so sour when you were young are not as sour as when you get older. You also have a number of olfactory receptors that declines with age. Now, do remember that these can be maintained a bit more if you actually use them and practice them over time, like a professional taster or chef. Gustatory reception is the process is stimulated by dissolved chemicals, and about 90% of the receptors respond to 2 or more different taste stimuli. There are different tastes involve different receptor mechanisms, but overall, the activation of the the gustatory receptor cells will stimulate the sensory nerve fibers that will then be relayed by one of these three cranial nerves, cranial nerve 7, 9, and 10. The facial nerve, cranial nerve 7, innervates taste buds on the anterior two-thirds of the tongue from the tip of the line of the velate papillae. Cranial nerve 9, or the glossopharyngeal nerve, innervates the velate papillae and the posterior one-third of the tongue. And finally, the vagus nerve, cranial nerve 10, is going to innervate taste buds on the surface of the epiglottis in the throat. And this would help to, of course, detect water and thirst. That's going to be it for this lecture. In the next lecture, we'll talk about hearing and equilibrium.